So while people are getting settled, I just wanted to say a few words. Thank you all. Good morning. My name is Ingrid Katz, and on behalf of myself and my co-chair here, Dr. Doug Kwan, we want to welcome you all to the Harvard University CFAR meeting. It is the 20th annual meeting, and we are so thrilled to be here, and it is an honor to have you all. I know many people have flown and come from a long way. Many of you have extremely busy schedules, and it is really incredible that you've made a little time to come and join us today. I think it's going to be a really exciting session. Our theme for today is Getting to Zero, What Will It Take? And we just wanted to give you a quick orientation to the day's events. We have two absolutely phenomenal, world-renowned speakers as our plenary and keynote speakers to bookend the day. So we have a first and last session, so that hopefully that will encourage you to spend the whole day with us today. The remainder of the format is comprised of brief oral presentations by our emerging scientific leaders in HIV. And then we will be having panel discussions by senior investigators, many of whom are mentors to the people who will be giving the oral presentations. So it'll be a nice exchange of ideas. And we are expecting and hoping for those panel discussions to be very robust and engaging. So we hope that you will join us for the day. We've tried to keep a very broad coverage of this area. Obviously, there are so many topics to cover, from sociobehavioral research all the way to basic science. So we've tried to keep a broad array of topics that we hope will keep you engaged. So before I hand over the mic to Dr. Kwan, who will introduce his mentor, I just want to give a special thank you from both of us to Mark Ingaciola and all of the Harvard University CFAR programming committee who've just done such a phenomenal job, and of course, most importantly, to the NIH for sponsoring the CFARs, which really are a continual gift for all of us who do this work.
Thanks. Well, on a day in which we're highlighting so many outstanding trainees and mentors, it's a real pleasure for me to introduce Bruce Walker, who served as my mentor since I've come to Harvard about 10 years ago. Bruce is the co-director of the Harvard CFAR, as well as the director of the Reagan Institute of MGH, MIT, and Harvard. As many of you know, he's been a central figure in the HIV field for decades and is known to be a truly generous leader, a gifted teacher, an inspirational physician scientist, and I suspect he may have somehow played a role in the Cubs win last night. So good job, Bruce. He really can do no wrong. He's someone whom certainly I owe an enormous debt to for his caring and tireless mentorship over the years. So with that, please let me welcome Bruce Walker.
Thanks, Doug. It was a late night last night, I think, for a lot of us. It's really my distinct pleasure to be able to introduce our first speaker, Dr. Anthony Fauci, who's the director of the National Institute of Allergy and Infectious Disease, and also is an inspirational leader of his own basic science research laboratory, the Laboratory of Immunoregulation at the NIH. I think it's fair to say that there is no single person in the HIV field looking back over this epidemic from the very beginning that has done more for combating the epidemic and for facilitating our lives as investigators than Dr. Fauci. And what is really impressive about this, I think, is that Dr. Fauci has always had the focus on patients. He's a clinician scientist extraordinaire who came at this from being a medical doctor and caring for patients, started out studying Wegener's granulomatosis and other inflammatory diseases, and when HIV came on the scene, he shifted his attention to that and has really never looked back. From the work that he's done in his laboratory, from the beginning of showing how immunoregulatory networks were influencing HIV, to studies of treatment and prevention, to more recent studies of intervention that actually show, I think, the greatest promise of anything out there right now, which he'll be talking about. He's just done a phenomenal, he's given us all a phenomenal gift. In addition to that, he is really the person that's responsible for all of us being able to be here today by supporting the CFARs over the years. When at times there has been question about the utility of the CFARs, I think all of us that live and breathe within the CFARs realize what a difference this program makes, and we're eternally grateful to Tony for supporting this. I think we also have to recognize the key role that he played in establishing PEPFAR, which has really changed the epidemic on a global scale. So without any further ado, let me ask Tony to come up and speak to us. And thank you so much, Tony.
Thank you very much, Bruce, for that kind introduction. It really is a pleasure to be here with you this morning to kick off this very important meeting of the National CFARs. As you see from the title of my talk, I'm going to focus in on the whole issue, which is of great interest to many, if not all of us in the room, is the issue of HIV persistence, and I'm going to look at it from a new approach that we have been working on in our laboratory for the last 10 years. So, information that you already know. This is a paper from my lab back from 17 years ago when, as a number of labs were concerned about when you have someone who's been suppressed in their viral load by measurements of plasma viremia for several years, if you stop therapy, do you indeed have persistence of suppression of viremia? And as we all very disappointingly found out, and this was one of the first studies that did that when you empirically discontinue therapy, the individuals invariably rebound to about the place that they were in their viral set point. We tried, as others have, to, quote, flush out the virus. And in this study, what we did is that we gave individuals who were suppressed on ART interleukin-2, which Cliff Lane had been working on for several years in the laboratory, to essentially activate the immune system. We could not find virus that was replication competent, although we were able to get some PCR out of them, and we did biopsies of gut, lymph node. We thought we may have actually essentially, I hate to use the word eradicate, but we naively thought that maybe we did that. When we stopped therapy, invariably in red, the virus came back, but more importantly, in the blue bars, the reservoir reestablished itself almost immediately following the rebound of virus. So how do we address HIV persistence? Let me very quickly go over this in introduction to where we're going with the main part of the talk. The two major approaches that labs throughout the country and the world have taken are shown on this slide. The first is an attempt to eradicate the reservoir, what we consider as the classic cure. There have been a number of attempts listed here. I will not go through each and every one of them, but some have been promising, others have been very disappointing, but none have yielded results that we feel are the kind that we could depend upon to truly eradicate the virus. The second one is one that my lab and a number of labs throughout the country and the world have been focusing on, is the issue of not eradication, but perhaps controlling viral rebound following cessation of antiretrovirals, what I refer to as sustained virological remission, similar to what people talk about with hepatitis C. Now, the paradigm that I've used in the laboratory over the years is outlined on this slide. To the extent possible, treat people as early as you can from the time they get infected. Take one of three approaches shown on the second line down, and then at an appropriate time, discontinue ART, hoping that you will then have sustained control of HIV viremia. So let's go quickly through that. The first is, if you suppress someone long enough, do they have enough natural residual HIV-specific immunity to prevent rebound? I just showed you our slide from our paper from 1999, and invariably that has not occurred, with some exceptions like the Visconti study, In our lab, we've not found the results that the Visconti study has had. I've discontinued therapy in patients who were started early, who were started intermediate, who were started late, and I've never had that kind of a percentage of sustained viral control. The next is therapeutic vaccination. We are in the process, and this is interesting, maybe sometime at the next CFAR meeting I could present this. We have a double-blind, placebo-controlled trial of therapeutic vaccination in individuals who get control, who get vaccinated, who've been on ART for years, and we've discontinued therapy. The results look interesting, but I don't know who's who because it's still double-blind. We'll know the answer in February, and hopefully we'll tell you a little bit about that. But what I really want to focus on is the issue of the passive transfer of antibodies, first done with the broadly neutralizing antibodies. And as we know, there are a number of broadly neutralizing antibodies, only a portion of which are shown on this slide, some of which have been approved for use in humans, such as VRC01, 3BNC117, and others. Michel Nutzensweig recently published in Nature that when he passively transferred 3BNC117, that he delayed viral rebound following cessation of antiretroviral. We did a study in our lab, and it's published literally in the next few days to a week, using VRC01, and it's a combined study between my lab and Pablo Tebas's lab in Penn, which showed the same sort of thing, a somewhat delay in rebound, but everyone still rebounds. So where does alpha-4 beta-7 come in on this? So let's take a quick look at this, because it goes back 10 years or longer in my laboratory. We had been studying over the previous 15 to 20 years the role of HIV envelope in the induction of aberrant signaling. And this slide schematically diagrams some of the things that we found. Mostly, when you trigger, you give aberrant signal, which makes the cell permissive to HIV infection. These are some of the studies that we did over the years, dating back from original study done by Drew Weissman when he was a fellow in my laboratory, in which we showed that HIV envelope can induce aberrant signals in CD4 positive T cells, which led to two things: dysfunction and permissiveness to HIV infection. Now, we did that with CD4 cells and monocyte macrophages, but we also knew that there was some NK cell dysfunction in HIV viremic individuals. So we triggered the cells with envelope, and we found we could induce that dysfunction. The problem is, as we know, NK cells lack surface CD4, and they have very low levels of CCR5. So what was it that the envelope was binding to on NK cells? To make a long story short, it was alpha-4 beta-7, an integrin whose natural ligands are MEDCAM, VCAM, and fibronectin, and who, as we know, its principal function is to mediate migration to and retention of leukocytes in the gut. So, we know it's a homing receptor. We know the gut plays a major role because of the bulk of lymphoid tissue. So it became very interesting to us that alpha-4 beta-7 could serve as another receptor, as it were, not necessary for infection, but certainly a receptor that binds to and triggers the CD4 positive T cell, and we'll get to CD4 in a moment. Now, we also know that primary HIV infection is associated with a preferential depletion of CD4 positive lymphocytes from effector sites in the gut. So obviously, when you have a molecule like alpha-4 beta-7, that's the homing receptor of cells that go to the gut in HIV infection, it became very clear to us that this is something that we wanted to pursue. So the next question was, does GP120 actually bind to alpha-4 beta-7 on CD4 positive T cells? And after a couple of years of work on that, we published it in 2008, 8.5 years ago, that in fact, the envelope does bind to, and it actually signals through the integrin alpha-4 beta-7 on peripheral CD4 positive T cells. Now, if you look at the conformational structure of what alpha-4 beta-7 looks like, it's closely associated with CD4 on the surface of the cell, and binds both CD4 and alpha-4 beta-7 when the envelope comes into close approximation. So here's some of the very quick studies showing that it actually does bind to both CD4 and alpha-4 beta-7. So in the blue is envelope binding alone. In the green is when you block the CD4 binding, so it goes from blue to green, but there's still some binding which you can further block with an antibody to alpha-4 beta-7, in this case HP2/1. The natural ligands, such as MADCAM and others, that bind to alpha-4 beta-7 bind through a tripeptide loop that has a central aspartic acid core. And when you do alignment of consensus sequences, you find that that tripeptide sits right in the V2 loop of GP120, which again became of considerable interest to us. So we continued to pursue. We then showed in a subsequent paper a couple of years later that alpha-4 beta-7 forms a very close complex with CD4, and it defines a subset of T cells that are highly susceptible to infection by HIV-1. And this is some of the data from that PNAS paper. If you see on the left panel, the beta-7 high cells are those that are preferentially infected at any level of viral input. So the dark bars on the right are B7 high cells compared to the light blue, which is B7 low or negative cells. Also, alpha-4 beta-7 high CD4 positive T cells are a preferred target for productive infection. Again, blue being high cells and red being low. And then we went on to show that if you look at the relative binding of founder-transmitted viruses, they bind much more readily to alpha-4 beta-7 positive cells than do highly glycosylated viruses that are obtained from someone after years of infection. Now, as this was going along, a number of other investigators were demonstrating that alpha-4 beta-7 positive T cells are targeted in acute infection. And these are just a few of the many papers that have been generated now with continued interest in this area. We were particularly struck by a few, one by Mario Roederer, showing that a high percentage of memory CD4 positive T cells are infected by SIV at peak infection, and CD4 positive T cell loss is wide across a wide spectrum of host tissues, not only the GI tract, but all peripheral tissues. Then what happened is that we noticed the paper that was done by an old friend and collaborator of mine from literally 40 years ago when he was at the National Naval Medical Center across the street from the NIH, Tab Ansari, when he was with the Naval Research Center. And he had shown that if he blocked in a non-human primate model alpha-4 beta-7 and acutely infected the animals, he saw a decrease in plasma and gastrointestinal tissue viral load, which was very interesting because it was an animal model validating what we were seeing in human ex vivo studies. So we decided that we would begin a collaboration with that group. And the first question is, can a monoclonal antibody directed against alpha-4 beta-7 protect against mucosal transmission? So what was used was a primatized antibody, a mouse primatized antibody, against alpha-4 beta-7. And I bring to your attention that an antibody to alpha-4 beta-7 exists and is commercially available called vedolizumab, which is used successfully in the induction and maintenance of therapy for both ulcerative colitis and Crohn's disease. We'll get back to that in a moment. So the first collaborative study, and I'm going to show you the data on that in a moment, was we asked, can antibody by passive transfer actually block transmission and perhaps protect cells from being infected? So here was the protocol that was used using a multiple vaginal challenge with SIV MAC 251. Infused alpha-4 beta-7 antibody or control immediately before or following low-dose vaginal challenges. And under the protocol, animals were challenged weekly, as shown in the green arrows, until 10 of 12 controls became viremic. And what we found was really very interesting, comparing the treated on the left and the control on the right. First of all, half of the animals who got anti-alpha-4 beta-7 did not get infected. Those who did, if you look at the onset of infection at 5+ weeks versus 2, 3, 4 weeks, the infection was delayed. If you do a Kaplan-Meier on that, you see both of these phenomenon exemplified. One, there's significant protection, and two, in those animals that did get infected, there was a delay in viremia. We also found something very interesting. If you looked at proviral DNA in the gut biopsies of the animal, now these are the animals that actually got infected. These are not the half that didn't get infected. The half that got infected had markedly decreased viral load in gut biopsies, and if you looked in other tissue, it wasn't just the gut. There was decreased virus in all tissue, but most pronounced in the gut. And if you looked at what other effect we saw, it minimized, as you see in the slide here, that if you look at CD4 cells, naive cells, central memory, and particularly effector memory cells were preserved in the animals in blue who got the anti-alpha-4 beta-7 compared to those in red who got the control antibody. So if you want to summarize what the major findings of that study were, obviously, as I mentioned, there was decrease in transmission following repeated challenge. But to our mind, even more importantly, is that there was a sustained decrease in proviral load in gut-associated lymphoid tissue, as well as other peripheral tissue following the termination of anti-alpha-4 beta-7. So then we asked the question, which is the subject now that we're pursuing very actively. Can infusions of anti-alpha-4 beta-7 induce sustained virological remission following discontinuation of antiretroviral therapy? So let's get back to my original paradigm that we've been using for the last several years, and where it says passive transfer of HIV-specific neutralizing antibodies, just substitute passive transfer of anti-alpha-4 beta-7 antibodies. And that's what we just recently published a couple of weeks ago in Science, in which we showed sustained virological control. So let me go over those data with you. The design of the study is as follows. 5 weeks of infection of the animal, followed by an additional 13 weeks of antiretroviral therapy, which overlapped at week 9 with an additional 23 weeks of anti-alpha-4 beta-7. ART is discontinued at week 18, and at week 32, all therapy is withdrawn. Sounds a little complicated, so maybe simplify it. Phase 1, infect them. Phase 2, treat them with antiretroviral. Phase 3, add either anti-alpha-4 beta-7 or control. Phase 4, get rid of ART. Phase 5, get rid of everything and follow them. And here are the results. If you look on the right, what you see is that viruses replicated well, the animals got infected, ART in gray made them aviremic. When we stopped antiretroviral, they all rebounded right back to where they were in the original 5 weeks of infection. If you look on the left, the same thing. They get infected. They are virally suppressed with ART. When you discontinue, 2 of the animals never rebound. 6 of them give blips that go up and down, and then they settle in and stay suppressed. So let's take a quicker look at that. These are the 2 animals on the left that never rebounded throughout the entire period of time. These are the animals that had the little blip. When you look at the geometric means of the plasma viremia levels in the non-human primates treated with ART, and look at the comparison between the anti-alpha 4 beta 7 and the control, the results are very clear. Now, this is out to 50 weeks. The studies were done in 2 sets of animals that were staggered by anywhere from 8 months to a year. The animals that were originally done are out now approximately 2 years following discontinuation of therapy, and the second group of animals are out about a year, and again, still being followed with no substantial rebound. It isn't a cure because you can get replication-competent virus out of the animals. They're just not rebounding with viremia. So let's take a closer look at what else is going on. There's individual proviral DNA in the gut. You see on the left, it goes up, goes back down, comes up, and then levels off, whereas proviral DNA in gut biopsies on the right in the controls stay there. This is the geometric mean of the proviral load in the gut of the control versus the treated animals. Let's just take a look at what else is going on, because as you're going to see in a minute, I'm not really sure what the mechanism of this is. So the kinetic changes. Total CD4 positive T cells, blue is treated, red is control. You get reconstitution of total cells, naive CD4, central memory, and interestingly, always better with effector memory than with other CD4 positive T cells. If you look at the gut CD4 positive T cells by biopsy and look at proviral DNA, you see again if you look at biopsies and identification by staining, which I'll show you in a moment, you see again total CD4 positive T cells and particularly effector memory CD4 positive T cells. Again, highly significant changes as you would expect, because the animals in blue are aviremic and the animals in red have continued viremia. So this is a biopsy of the gut comparing the control on the left with the treated alpha 4 beta 7 antibody on the right, and this is an immunohistological staining of CD4 positive T cells in the gut biopsy specimens. We don't know whether this is an effect of what we're seeing or whether it's a potential cause of it or a combination of both or has nothing at all to do with it. So what are some of the other peripheral observations in non-human primates who received the anti-alpha 4 beta 7? Well, there was an initial loss, then a recovery of Th17 and 22 subsets of CD4 positive T cells. There was a significant increase in cytokine synthesizing NK cells in the blood and the gut. And that's interesting because Veffa-Vancini did a study in which she tried to recapitulate RV144 in a non-human primate model. And in the protected animals, she saw that too. Whether that has anything to do with cause-effect, we don't know. There were plasma biomarker signatures associated with reduced inflammation and gut damage. And then there was a recovery of reduced retinoic acid levels. Retinoic acid is made in dendritic cells in the gut, and it actually upregulates the expression of alpha 4 beta 7 on CD4 positive T cells. It initially went down and then it normalized. Whether that has just peripheral, parenthetical effect of damage of the gut not being as severe, we don't know, but that's just the observation. The other interesting thing was the frequency of NKp44 subset of innate lymphoid cells in the gut went way up in the individuals who were treated with the anti-alpha 4 beta 7 and maintained low in the control animals. Again, not sure what this means, but this was an observation that was made in a vaccine non-human primate study in which there was some protection in that study by Veffa-Vancini and her colleagues. So what about antibodies? The geometric mean of antibodies to GP120 as a whole molecule was no difference between the anti-alpha 4 beta 7 and the IG controls. We looked for ADCC and neutralizing antibodies, and we didn't find any substantial difference between the two groups regarding ADCC or neutralizing. However, when we looked at specific antibodies against the V2 loop, we found something interesting but unexplainable, is that 3 out of the 7 individuals, monkeys, who got control had antibody to V2, whereas all 8 of the animals who got antibody to alpha-4 beta-7 developed antibody against V2. Now, all of a sudden, that's a big red flag when you're thinking about antibodies to V2 for the following reason, is that the only trial of a vaccine that has any signal of efficacy was RV144, and when Bart Haynes and others looked at correlates of protection, they found, as we all know from papers from 4 years ago, IgG antibodies against V1, V2, particularly V2, associated with reduced infection. They were non-neutralizing, but they did mediate ADCC, and they had a contrary effect with IgA. Now, when Roland and Haynes did sieve analyses on the antibodies that were made in the RV144 correlates, they found that they selected out a particular stretch of amino acid on the V2. So we did a similar type of a study. We did competitive studies against antibody binding to the envelope by overlapping 15 mer peptides to see where the antibodies to V2 were actually located. And we saw the most profound inhibition at peptide number 43. And when you align peptide 43 with the same stretch of the virus that was used in the vaccine for RV144, there's a highly significant match. Again, the significance of that, in my mind, is completely unclear. It could be pure happenstance, but it really is interesting that they induce those antibodies. So just to summarize what I said, it's of interest that the SIV macaque 239 V2 peptide from our study overlapped with the site of antibody immune pressure in the V2 that correlated with decreased transmission in the RV144 vaccine study. And as I mentioned, the significance of this is unclear. So in summary, infusions of anti-alpha-4 beta-7 together with a 90-day course of ART initiated 5 weeks after infection resulted in long-term virological suppression. CD4 positive T cells were restored in the gut, but also in the blood and several lymphoid tissues. The mechanisms are unknown. I've already listed what I saw as observations. Whether they are significant in any manner or form, I don't know. But one thing, that these studies certainly may provide insight into potential mechanisms that might be pursued for the prevention of HIV infection and or the maintenance of sustained virological remission. And in that regard, we've already started an exploratory open-label study of vedolizumab in subjects who will be undergoing analytical treatment interruption. That's the clinicaltrials.gov number. The FDA is very concerned about safety, so they're making us go very, very carefully. It took us about 3.5 months of negotiating with the FDA about what the specific aspects of the protocol were going to be when you reinstitute following rebound. We have permission to do as few as 15 and as many as 20 to 25 patients. We've enrolled them already. There are 3 already in the study, 4 lined up to go in the next couple of weeks. Hopefully, we will have it fully enrolled by the next several months. But remember, the answer to this is going to take many months because we have to treat the individuals for at least 4 months with antibody to alpha-4 beta-7, discontinue, and then follow them for another several months. So I don't think we're going to get an answer to this, whether or not it's relevant in humans or not, for at least as we get well into 2017. So let me close by thanking my collaborators, who are many in all of this. Remember, this was a combination of human studies that were culminated in animal studies. But I want to go specifically to give credit to these individuals who are very terrific collaborators, particularly Tal Bansari's lab, my old friend from way back at his time at the Navy, and his very good crew at Emory. the German Primate Center with Lutz Walter and colleagues, the Yerkes Primate Center with Francois Villinger and his colleagues, Georgia Tech with Phil Santangelo, the University of Manitoba with Lyle McKinnon, Hopkins with Raffaello Cimbro, Michigan with Kelly Arnold and her colleagues, the Department of Pathology at Emory with Tristram Parslow, the University of Maryland with Maureen Kane, and finally my own group at the NIH immunopathogenesis section in my lab, particularly Jim Arthurs and Claudio Cicala. Thank you.
So we'll go ahead and take some questions. Maybe I can start. So in the monkey studies, they were early treated monkeys. Do you think that has a role, and how are the patients being selected for your clinical study?
Yeah. So you have to be careful. Great question. So we treated the monkeys for 5 weeks, which is longer than the usual studies that treat monkeys. So it was kind of like not acute, but it wasn't chronic. It was somewhere like subacute. Now, the interesting situation is the question going to be, is it critical to give this if it works in humans? Would it be critical to give it at the time that you start therapy in an acute patient versus a patient that's treated acutely that you do the alpha-4 beta-7 2 years later versus someone who is treated chronically and you give it? So there are 3 separate groups. We don't know, A, whether it's going to work in any of them, and B, if it doesn't work in chronic, obviously you're going to want to get patients that not only are acutely infected, but treated with antiretroviral and antibody to alpha beta-7 during the early phase. So the FDA wanted us to work with stably controlled HIV-infected individuals because they didn't want it. They were really very concerned, understandably, any of you who work with the FDA, about safety. So they say, we want patients who are really stable before you do anything with them.
Let me ask one quick follow-up question, then we'll go to John, and that is that it's fascinating data about the DNA in the reservoir. What do you think that says about establishment and maintenance of the reservoir?
When you say the fascinating what? That it went down?
That it went down so much.
Yeah. Well, I think that the reservoir is, maybe people can disagree with me, is really quite dynamic.
This is a place for disagreement.
It's quite dynamic. I think it is not just stable. Yeah. It's quite dynamic, and you could perturb it in ways. We've not been able to get rid of it, but you could really perturb it. And we showed that 12, 13 years ago when we blasted away with IL-2. We couldn't find any reservoir, but it obviously was there because everybody rebounded when we stopped.
John?
Thanks for that inspiring talk, Tony. 2 quick questions. One, is the integrin expressed on the surface of SIV? And have you done any binding study to see whether the antibody is mediating clearance or FC receptor mediated effector functions? And second, the primatized antibody has an intact FC domain. Does vedolizumab?
The primatized has an FC domain. Yes, it does. It does. Right. So let me answer your question. I live in Washington, D.C., and as George Washington says, I cannot tell a lie about the cherry tree. The answer is alpha-4 beta-7 is expressed on virus when it buds out. Right. That may be one of the mechanisms.
But I heard the FC domain of the clinically approved product vedolizumab has been modified so it doesn't mediate the same effective function that the primatized antibody. Is that correct?
I'm not sure. I'll have to check. I don't know, John.
Thanks.
I don't know.
So, Tony, I may have missed it with all the data you showed, but the question is with the reduced DNA copy number per million CD4s of various subsets. Is the apparent reduction per million due to the fact that you've actually reduced the numerator, or because you've got reconstitution or generation of a large number of cells in the tissues and the periphery of CD4 cells,
Yeah.
that the denominator is increased, and so that number is lower.
That's a really good question, and I think that could be a factor, Doug. That's quite, in fact, I brought that up with you. See, as you well know, I'm not a non-human primate person. I didn't do those biopsies, but that was the first question I asked. So you really have such reconstitution that you just diluted it out, and it's the same amount. It's just, it looks better because there's more normal there, right?
Savita, and maybe we could have people, when they ask questions, just identify where they're from.
Yeah, I'm Savita Power from University of Miami. Thank you for that lovely talk. I'm curious about the V2 antibody. Do you think that was induced, or why did it increase?
Well, it had to be induced because it wouldn't have increased. I don't know why that happened. I mean, you could speculate a lot and say that somehow or other, the antibody bound to an infected cell through the alpha-4 beta-7 on the cell or on the virus or something and presented itself to the immune system in a way that natural infection doesn't, which then allowed you to get an antibody against V2. That's possible. I think it was induced. My question, Savita, is less was it induced, which I think it was, is does that have anything at all to do with the protection, which I don't know.
Because it doesn't have ADCC activity.
Right. It doesn't have ADCC.
Thank you.
So there's a lot of unknowns about this. The unknown that I wouldn't say troubles me, that I always act very measured and reserved about, is that being a human HIV person, I'd like to see it work in the human before I get even slightly excited about the results in an animal. Right.
It's very encouraging.
Tony, fascinating. I'm Jim Hoxie from the Penn CFAR. So Tony, with the animals that you have that are out a year or so and have the state that you've described, so you could do a lot of things with those animals. Have you thought about things like CD8 depleting them, challenging them, to sort of get at themes of what really is controlling this?
Okay, so that's a great question, Jim. Tab Ansari has attempted to CD8 deplete, but the experiments technically did not work. They were not complete CD8 depletion, so he's repeating them. So the answer to your question is yes, he's trying to find out what the mechanism is by CD8 depletion, but he hasn't done it yet to the extent that he could say it does or does not. But that's the first thing that you would think of, is it inducing a CD8 phenomenon? Which would make the most sense. You know, that kind of control would much more likely be cell-mediated than antibody. Oh, absolutely, yeah.
Great. Thanks very much.
You're welcome.
Thanks, Tony, for getting us off to a really terrific start. We'll move on now to panel 1. The moderators are Carl Diefenbach from the NIH and Doug Kwan from the Harvard CFAR.